Testing 1 2. Testing 1 2. Alright, just gonna get some audio levels here. Gotta lift up screen here too. Hold on a second. I had to redo my OBS settings. There were some issues. So still seems a little squashed, but I'll just leave it that way for now. The slide is going to dry out on me. Chat so we can see it better. I think I have the music too loud again. You can barely hear me, or it's similar volume. Just a tad louder, I think we'll fix that. Music's too low. I had all this stuff dialed in last time I streamed, but if you guys, I'm sure you noticed I haven't had time to do this in a while, so. Okay, I think that's a good match right there. Alright, start with that. So I was at my nephew's wedding today down in Carmel near Point Lobos. On the way down there, it's a place called Monastery Beach. Beautiful place, great for scuba diving, scenics, lots of weddings done down there, it's general photography, and normal beach or lounging activities. So I grabbed some samples out of the water real quick before we had to leave. And I got some diatoms here. Zoom in on this guy. A little digital zoom just to help me out here. Alright, so see two diatoms there. One on the top by the air bubble. It's not as structured as the other one, so it may be dying or on its way out. But the one down below is a nice, perfectly filled rod. See plenty of chlorophyll in there and stuff. These kind of diatoms have that kind of more of a golden chlorophyll, not the bright green you get to see in a lot of other plants. And to be honest, it's been a while since I looked it up, so I need to look that up again and see what the chemical difference is. But you know, those diatoms generally have the golden look, more than a green look. I need to. What's up, Matt? How's it going? Thanks Thanks for the birthday greetings. Yeah, last week. Big old birthday. Getting older and older and older. I was actually out of town at that time, so I had, what did we do that day? I'm trying to remember what we did that day. I think we're in Universal Studios that day. I jumped in on Kent's pub. I was making fun of Harry Potter stuff and all that. The signal sucked. I think that was my birthday. I can't remember. I have to go back and look. So I'm just going to do a quick check. The focus seems to be off here a tad. Sometimes the adapter has some drift, and so what's on the camera is not the same as in my eyepieces. That's a little closer to what I should see. That way I'm only fiddling with the scope a lot and less on the software side. And is that my field of view? Kind of. Zoom out a little bit more. That's more like my field of view there. Okay, cool. So let's go in one more level. Let's see. Nice diatom there, and I gotta fix the brightness on this guy. Get that cursor out of there, it's annoying. It's useful at times, but it gets in the way. So really nice view of a pretty good sized diatom. See some of the little things bopping around it with some Brownian motion. The heat of the lamp is making the water molecules dance. There's actually some, looks like some very large bacteria there bouncing around. If I tweak the this a little bit, it might help you guys see it. Or just go to dark field. Usually dark field is better for the camera to see the tiny bits. Let me try dark field. I don't know if this one is configured to go dark field. Let's see. I got some oblique light in there a little bit. Let's get a little more contrast. Sometimes. Alright, so you get some little bit of a 3D effect there. There's a little bit of oblique lighting. Somewhere between bright field and oblique lighting. You can maybe make out those little guys bouncing around there. If I defocus them, it shows up a little better. There's a little sphere there kind of bouncing around. So yeah, not a lot on this slide, to be honest. I've had a couple diatoms so far. Let's go back up, look at this other diatoms up here. There's one here. You can see the salt crystals are starting to form already in this marine water. There's a diatom. Looks like it's falling apart there. There's a diatom here. You can see there's kind of a shrinkage. The material, it's pulled away from the little glass shell that makes the outline there. So this one could be either dead or dying as it's shrinking instead of being fully extended throughout the entire shell. Make sure I get this aligned correctly. It's still not quite aligned. this over. That's more like what I'm seeing right about there. So again, diatoms are always cool, but they're, after a while, they're fairly dull, because in this case, these aren't even moving. Usually they're at least slowly moving around. But I'm not seeing any of these move. So that was a good starter slide. Let me go to another one. We're already getting ice crystals. Salt crystals on that one, because the slide was sitting there for a while while I was messing around with the stupid OBS settings. Let me make another sample. So what I did is I went got some ocean water in the bag, some sand, and some beach rack. You know, some of the pieces of dried kelp and stuff that often have little critters of all kinds, and I just kind of tossed them in a bag because I only had one bag with me. And I'm making some preparations from that single bag. In fact, I'm seeing already some of the red algae that I collected on the beach, probably someone called Mastocarpus papillata, or Turkish towel. Used to be called Gigartina, but they changed the name many years ago when I was studying marine algae. Had to relearn stuff all over again. And it's a cool looking algae because it really looks like a very coarse towel you would have in a Turkish bath, or at if you choose to buy that kind for your home at a home goods store. Now I got some dark field here. This might help a little bit. Find some more goodies. Got some nice more JX3D space music here in the background. Go get his stuff on Bandcamp, JX3D. I finally put up a Bandcamp channel. I think Matt's stuff have been on there, so I appreciate that, Matt. If you guys want to check out stuff, but this is all JX3D and there's not many of my music, but it's cool with as the particles are going back and forth, you can see zoom in just a tad so I don't have to change my patch stops on here. And get some spacey music with particles drifting slowly through the field. Find a better drifting section here. I think they all equalized out now. It's really just a equalization of the gradient, basically some more salty parts and less salty parts, or warmer parts, colder parts, whatever. So you get this equalization as particles shift around to an equilibrium state. Getting a lot of... That's cool. You can see the edge lighting on those. Looks like some diatom tests that are gone. They're just empty. So they're definitely not your normal air bubbles. They have the oval and kind of rounded rectangular structures on the left side there. They're getting some edge lighting right here and right here. Actually, was that showing up okay on that? I think I'm pointing stuff out on the wrong window. Hold on a second. There we go. So yeah, right here and right there. It's kind of cool little outlines there. Zoomed in. Not see anything alive on here, at least at this scale. And again, I just grabbed some ocean water as it ran up the beach in a bag. Threw some beach rack in there. And some sand. I want to take some of it and look at the dissecting scope in a bit. See if any macro stuff is crawling around in it. That's why I only put so much water in the bag. I didn't want to drown everything that wants to be on the sand, crawling through the kelp. Let me try that real quick. I have a nice little petri dish here. I'm going to grab some bits of beach rack. Random stuff. Even a gas bladder from one of the kelp. A lot of the gas bladders in kelp have some different compositions. Some have like carbon monoxide, which is kind of interesting. The same stuff you get out of your car when you're combusting fuel. It's unfortunate people use it to commit suicide sometimes and other things with it. So it's definitely not a compound you want to be around. And I can't remember. You read about it years ago. More than years ago, decades ago. And I think it's just a clever way for the plant to make a waste product and store it in a place that's not affecting the biology of the plant, but also gives it flotation. So it's a win-win. Just like how your liver stores nasty products, so you don't die from various things within reason, which is why alcohol has that effect on your body. You get alcohol dehydrogenase in your liver, breaks it down until you just drink yourself like a fish, and then you don't have enough, and then you start getting some damage. A lot of body processes create some other nasty products or side products, and if you can't excrete it safely without hurting the body, sometimes you get sequestered in the body. So let's switch cameras here to the dissecting scope. We have some focus issues right away. Oh, come on, this cable's too short. There we go. Alright. Now we're on a macro level. Zoom out a little bit more. I can move this tray around and turn this light down a tad. Is this back? No, it's not backlit yet. Let's see. Sometimes the backlit helps a bit first. Let's do all backlight. So you can see through some structures here. It's not working too well with that. Some of these are pretty thick. There you go, this is a nice structure, almost like a fractal shape there on the edge of this algae here. Let's go back to top light. Where's the gas bladder here? Here it is. There's a big old gas bladder for one of the brown. So you have brown algae, red algae, and green algae. Those are the three main types in the ocean. So, what is it? Chlorophyta, Phaeophyta, and Rhodophyta for the red. So this is Phaeophyta. Those are the major groupings. So this hasn't been punctured yet. It's starting to dry. It's been on the beach for a while with that water. And Phaeophyta is PHAE. So you can see the stalk here called the stipe, and it has some little leafy structures hanging off. They're not actual leaves because this is a type of algae. And the gas bladder comes off one of the branches, and you continue to go up through the oops, knocked my light off. Hello. Hey. Come on, light. Oh, I see what I did. I bumped the power supply's temperamental on this thing. There we go. So if you verify my focus on this, make sure, yeah, this is way off on the focus. And this stuff's pretty big, so I'm like way zoomed out on this thing. You can see these little, those little branching structures again. Go with a softer light on this one. This might help better. There we go. There's a little backlight again, so you can see the nice structure as well as some of the color as light is going through it. New backlight gives it a little more of a greenish tint, but it's really a brown. If I go back to top light, that's the actual color. And add a little extra backlight to give it a combo view. And what else do I see on here? There's some little micro chain bladders there coming off of it. So they're not just voids, they tend to have some kind of gas in it. The last times I was reading about all this stuff. I have to go back and do some more research before I do this next time. This is a quick and dirty stream with stuff I got while it's still fresh. Got home, got pretty tired, but wanted to share some of the stuff before it goes bad. And here's this, some of the sand. It's still wet. You see some quite large pieces of eroded minerals in the sand. All different colors, not just silica, silicon dioxide, it's all kinds of other colorful bits in there from different rocks that broke down in the area, became part of the sand. At a glance, the sand was kind of a brownish, light brown, reddish color, mostly brownish color. So for fun, I was going to take a little bit of the sand and put it on a slide, just naked, because I'm not going to be able to get close to it with the compound scope anyway. Put some of this on so we can get closer to it, because I can only get so close with the dissecting scope. Let's go ahead and check this out under the compound scope again. Let's switch back the camera. Got some problems here. Hold on a second. It's still dark. What did I do wrong here? Close down this too much. Oh, interesting. I thought it clicked in, but the turret was not fully clicked in. That's silly. Very silly. So you can see how these are huge under here. This is on the lowest power. So we're looking through one of the lighter pieces of the sand. You can see all kinds of it's transmission light, not reflected light. So you're getting all these inclusions in there and stuff. Like if you're looking through gems, but this is chunky, worn down minerals, not just silica, but a combination of stuff. All kinds of, it's heavily polished, like a lapidary, like you put in the tumbling machine and you get your nice polished rocks. Well, the ocean's doing that, you know, millions and millions of times to this stuff until it finally wears down to little sand grains. But right now they're pretty chunky. And they're all slightly different levels of polish and color or jaggedness, depending on where they came from, how long they've been in the matrix there. Now I can see the sheen from the moisture. It's got in water, and it looks all shiny. So, some edge lighting. Shiny little tiny rock, shiny tiny rock. And back to the transparent one, which is probably the coolest one here. That's just a sample of some of the coarse sand on the beach. See if I can find a couple more bits real quick for some variety. So just pretty chunky. So we get some more out of the bag directly. Ah, cool, you did some stuff in a soil lab, that's neat. We did soils lab in my ecology class in college. It was more about the organisms than the actual soil. We did talk about this, the levels of soil. I forgot that it's like O, A, blah, 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 these different letters for the different horizon levels. And I'm just drawing a blank on a lot of the terminology because it's been so long. But we would run the soil through, oh, here's some more right here. No, these are the same ones. I put some more on the side here, I thought. There we go, there's another one here. And the cool thing is you put them in this apparatus called a Tolgren funnel, and you put heat lamps on the top. And any organisms in the soil would migrate to the lowest levels based on their sensitivity to heat and light and all that kind of stuff. And you get this either a collection at the very bottom, or you get sometimes the stratification if you pick at the different layers in the sample you left in there. But most of them tend to fall toward the bottom. And then you have a nice little collection of insects that run the soil and other little critters without having to pick through with a microscope little by little by little, and also potentially destroying the insects with the little forceps and stuff. You just let them migrate through as they escape from the heat. It's like a soil version like a soil version of the famous dish they have in Japan where you put little eels in a pot and a big block of tofu, and you slowly turn up the heat. And the eels go into the cold tofu to escape the heat. Eventually it gets so hot, they die and cook inside the tofu. You can slice the tofu and it's like Japanese head cheese. So, it's kind of a gruesome thing. I would never eat it, even though I used to eat meat. But the principle is similar. You're driving them down from the heat source into another location. Anyways, that's a little bit of sand analysis there. Let's play with a few more macro bits here with the other guy. What are the samples we can get here? I think it's playing the same track. Is it playing the same track over and over again? It's supposed to add advance to the next one. I did some goofy setting on the loop, I think. Hold on a second. It's not supposed to keep playing the same track over and over again. Not that one. This one. Properties. Oh yeah. Okay, now it shouldn't keep playing the same track. I clicked the wrong loop button. Should go to the next one in the folder, hopefully. Because nice music is nice, but not if it's playing the same track over and over and over again. Whoa. Why did it change? I didn't tell it to change. Let me jump ahead a little bit here. See if it goes to another track. Now the software's confused. Come on. No? Oh, that's weird. I goofed that up somehow. Hold on a second. So what happens when I had to reset everything? I forgot how I had all these things dialed in. And I don't want to waste too much time on the stream getting it to work. Let me kill that. Let me check the properties on that. That's not a file. Oh, let's play it again. And now it's confused. Bear with me while I quickly switch to another source. Set to cancel. Oh, I see what it did. That's not an audio feature, that's a video feature. And this is Really exciting. Sorry about this. I thought I had this all sorted out, but it's... Let me kill that one. Start up another one. Application audio. Okay. I'm going to go to... there. Okay. I'll go back to this program and play it. Too loud. I just solved it. Okay, now I have to unmute this real quick to make sure I'm not stepping on. I think it's too loud. Here, let's drop it down. Oh, no, it's not doing anything. But it's definitely lower on the VU meter than my voice. Yeah, you guys have to tell me if that's okay. I can't. I'm getting duplicate audio sources now with the music with this other method, so I'm just gonna let it play in the background one way, and I have to mute the live feed. Just give me a holler if you're still there. If the music is the right level, you can still hear me, or if the music is overpowering, or I'm overpowering. What was I gonna do? Oh yeah, I was gonna get some new big samples here. See what else we can find in the bee tracker. This was a pretty warm day, and this stuff is pretty dried out. So I'm assuming even though it's rehydrated, it's not going to be great. But here's a beautiful shot of the Turkish towel. As I mentioned before, the red algae. So there's little bumps all over it, like a coarse towel. Cool, thanks, Matt. So this is Turkish towel, red algae, and you can see when I do this. Ooh, it's pretty dark. I can't even transmit through it very well. But you can see on the edge, see it's reddish. This is one of the Rhodophyta. This is the family you get a lot of the, sorry, not family, it's an order, I believe. I think it's an order, Rhodophyta. There's a phylum, Jesus. I forgot a lot of this stuff, I gotta go back and look. In any case, the red algae is where you get a lot of your stuff like your carrageenan and other kinds of algae products used in toothpaste and some ice creams. If you read your label, it says carrageenan. There's one called locust bean gum. That's a plant, not an algae. But carrageenan is very commonly used in toothpaste and certain types of ice cream as filler. You know, unless you get the more homemade style ice cream. So it gives you the texture and the viscosity and the mouthfeel that simulates full cream, homemade, heavy-duty ice cream. But carrageenan not only makes it cheaper to make, but also makes it lower in calories potentially, and you know, because it's not really it's not really a digestible thing. It's more like a cellulose type product. In any case, it's you know, farmed in large quantities, used for all kinds of stuff. They use them in other types of algae, used in paint bases, and all kinds of stuff. So you can use gelatin in some cases. You can use carrageenan. You can use other kinds of plant products for these types of things. Let's do this again with more light. So you can see some coarse sand all around it. These are pretty big chunks of sand. I'm going to zoom in a bit on this Turkish towel. And if I get maybe a little more light at a different angle, it might help show the shape of it a little bit more. So it's been out in the sun for a long time, but I picked up. It was still kind of soft and pliable, but it was definitely much drier than when it comes out of the ocean. And being in this bag with the ocean water, it's been rehydrated a little bit, but it's not quite the normal look it would have. But algae's pretty durable. See all the little bumps there, like papillae, like your taste buds on your tongue. So that's Turkish towel. One of the red algae. And then I've got some other algae mixed in here. There's more of that one here. I think this is, oh boy, now I gotta think about what this one's called again, with the chain bladders. Even looks greenish here, it's more of a brown algae. But remember, it's going through, that part's all kind of dried out and messed up, so this is not the normal color of this algae. But they can anywhere between a dark green to a dark brown, yellowish brown, like giant kelp called Macrocystis pyrifera. It makes the giant kelp forests off the coast here. I mean, it's basically underwater redwood trees, for lack of a better description. It feels like you're in the redwood forest if you're scuba diving, kind of cruising through them, looking up the shafts of light coming down through the trees. All the animals live at different stratifications in the kelp. It's beautiful stuff. If you can't get to an aquarium or you can't scuba dive, just go watch some really nice documentaries on the kelp forests. I'm sure David Attenborough has probably 4 or 5 of them. And it's just magnificent. So nothing like being there, but aquarium's next best thing, like especially the Monterey Bay Aquarium, because they have a massive kelp forest tank where it was really tall strands of it. So, and the shafts of light coming through there, and the little fishies hiding and eating. They have like turban snails in there eating their bits of the kelp, and crabs, kelp crabs, all kinds of stuff in there, all the different stratification, and just like a real forest on land. Kelp forest is really dynamic, really interesting place. And if you're also afraid of sharks in general, little leopard sharks go through there once in a while and stuff, but like you don't get the bigger sharks, and certainly not great whites. They stay out of the kelp forest. I've never seen them go anywhere near one, because their food isn't found in there. They're looking for larger prey. So not only is it beautiful, and you see all this life in different colors, and all different shapes and stuff, but you're pretty much shark free, except for, like I said, little leopard sharks, which are harmless and stuff like that. So it's a great place in so many ways. It's very peaceful and beautiful. But I didn't get any of that. None of that washed up that I could tell on the beach. So that's Turkish towel. If I had my microtome out, I'd do a little slice of this and put it under the compound scope, but that piece is way too big for the normal microscope. Let's see what else I can find here. I think this might be some eel grass. Yep, there's some eel grass. Piece of it. There. Let's get to magnification and cut down the light levels here. Here's eel grass. It grows quite long, and again, it grows like a long grass would grow, like in bunches, like a bunch grass in the wild, like in a grassland in where I live or lots of other parts of the world. And you know, it waves, like the grass waves in the wind as you go through the prairies and stuff. This waves underwater. Very beautiful stands of eel grass. And again, all kinds of critters crawl through it, swim through it, hang around it, eat it, all kinds of stuff. So it's a proper flowering plant. It's not an algae. This is just a piece of it. They grow quite long. And then let me grab some other stuff here. Bladder rack. I've not heard of that. Oh, you mean like the bladders on making the beach rack? Or is that a specific algae? Some more pieces of stuff. Oh, looks like I got a feather in here from something. Again, everything washes up on the beach, collects in the beach rack. So you get isopods, amphipods, feathers. Where's the feather? I just had it in here. Just had it. Where'd it go? Oh, here. Is it there? Yeah, right there. There's a feather right there. Get my forceps on camera. Everything's reversed. So there we go. There's your feather. Small feather from something. Very small feather. Do a little backlight on that. Kill this light. Yeah, beautiful feather there. It's all the barbs and barbules aren't working too well. It's been trashed around, so it's not zipping up 100%, but that's what it is. What else did I get in here? Some more this feathery brown algae. Pretty cool. Let's see what else I got in the bag. Start dumping more stuff out. So there's not a lot of standing water left in the bag since everything absorbed the water I did collect in here. And so far you just saw some diatoms and stuff, nothing too exciting. And sometimes you get that. It's just that a section of the ocean didn't have a lot of floaters in there, like dinoflagellates and stuff. Ooh, that's a big feather there. Some other little bits of stuff. Looks like a little piece of coralline algae. That's another type of red algae. encrusted with calcium carbonate, or chalk in one form. So, where's the coralline algae? Right there is coralline algae, so this red guy right here. If you pinch it, it's crunchy, it's very firm. It's essentially flexible, so it forms little plates, calcium carbonate, and gives the plants, the algae, some rigidity, but also pliability, because it's jointed. To zoom on that a little bit, it's pretty cool stuff. Some are more interesting than others, but just the fact that the red algae can do this, those little, almost looks like an insect leg or something like that with the little segments. But it's a coralline algae, forming little lobes and branching off different directions, and you get these little like it's very thin and flanged. See it in different directions. It's symmetrical and flanged, and you get these little pores on it sometimes. That's the algae where it's reproducing, most likely. Or the algae life cycle. One more light here. Looks almost like it has a disease or something like that, but you get these pores sometimes forming on it. It's usually their reproductive sections there. As far as I remember, again, this is decades ago. It's kind of a blur. I gotta go back and review. Just like anything else, you forget. But shapes are really cool. This would be great stuff to copy for like alien designs and stuff for 3D animation or cartoons or whatever. Like different exotic armor. Yeah, it has certain types of cacti, things like that, can have that same look. But they're pretty soft. They're just fibrous and waxy. This is actually firm because of calcium carbonate. And if you walk through tide pools and stuff with a bunch of it on there and you step on it, you'll hear it go as it's crunching against the rocks between your feet and the rocks. Alright, what else we got here? Almost done with the bag. I just want to go through real quick and just see anything else cool that pops out. Or this small brown algae. I just don't know this one very well. Used to know this with that Chainbladder. I've got to look that up. Pretty distinct. Let me zoom out again. There's the Chainbladder there. Some brown algae. Let's try backlight. What did that do? Kill that light, kill that light. Let's do more backlight. Gets a little bit of transmission through some of that stuff, but not too much. But if you look at the coralline algae, you might get maybe a little glow from it, but it's pretty opaque because of the calcium carbonate. Definitely can't shine the light through a piece of chalk. It's this right here. Here's a different bladder, one of those little tapered bladders. This looks like one of the bladders on the giant kelp. I'm gonna turn the light up here. Again, pretty big. It's hard to get it all in the frame. Oh, critter in there. See that? Somebody just crawled through the kelp. I brought a little visitor home. Probably a amphipod or something. Called a beach hopper. We also have little isopods and other stuff that live in here. It's basically like a little jungle for them, right? Plenty of stuff to climb on. Got moisture. You got other things to interact with. Plenty of stuff to eat. They can find other little things if they're carnivorous. They can find little things to eat. Where'd it go? I have to light it from below to see it better. I did see a little guy walk over the algae a little bit. Probably does not appreciate the bright lights. Let me clean this. I got junk on the bottom of this. Is making scrapey noises. Let's see where he emerges. Where are you? I saw you. And that's again why I didn't flood the bag, because there's some things in here that breathe air, living in the beach rack. I didn't want to destroy them. Take a look at them. Get a little more top light. Maybe help it see it better. Let me just let it sit for a bit and see if anybody moves across the frame. Yeah, if you study any science fiction artists or guys doing stuff for movies, TV, especially science fiction based stuff, or maybe some horror, they're totally grabbing stuff from nature and stuff, because why not? It was already there. You just emulate it and synthesize it into your work. Don't reinvent the wheel if you don't have to. That's me moving all that shit. Let's see if we can get... I might just have to go back and review the stream. That might be the only shot of the little guy. I did see a crawl over. They had an incredible exhibit years ago. At California Academy of Sciences in Golden Gate Park, one of my favorite museums. I grew up nearby. So my parents took me up there to school, go up there, and I got old enough to drive. I could just go up there myself. And I would spend hours and hours and hours in that museum. And around the time I got more into computers and started doing some stuff with graphics and stuff, they had a really cool exhibit called Life Through Time. It was all the latest information. This is way before Jurassic Park came out. But it had a creature essentially featured in Jurassic Park. It's not Velociraptor, it's Deinonychus. And very menacing looking with those little raptorial claws. And just, they look like crazy hunters. Velociraptor is actually much smaller than Deinonychus. But Deinonychus looks more menacing and cooler than Velociraptor does in real life, from all the images and skeletons I've seen, recreations of the stuff. So they had that in the exhibit. They had like giant centipedes that used to exist, pretty much to scale, in these like swamps that eventually became the Carboniferous period, which is where we get a lot of our coal and oil from. All those crushed animals and plants from the time period to create those deposits over millions of years. And anyway, so they have this thing you literally walking through time as you go to different rooms. So you have dinosaur periods, you have this primitive stuff, you have trilobites, you have these crazy diatoms. It's like these giant walking birds that look part dinosaur, part bird. Huge beaks on them, feathers and all that stuff. Really nasty. It's like, you know, later on you had like elephant birds and moas, things like that, which are just basically giant crazy ostriches essentially, but much taller. These are like nasty, like they're just gonna kill you, like something you see in a Ray Harryhausen movie or something. And then they have these computer terminals too, and you can go look at phylogenetic trees and clades of all these related organisms based on genetics and morphology and all the usual stuff. But a lot of the new information was on the genetics, which changed a lot of stuff. And I was so happy when about a year into the exhibit, maybe more, they started selling the software in the store. So on your Macintosh, you could take the CD-ROMs home of almost every computer exhibit you saw in their home, with all the trees and all the text, all the graphics and animations and stuff. It was like the coolest thing. No museum does that. They're not going to give away their assets like that, especially when the exhibit is still open. And I was just fascinated by it. Now, it was made in Director and ran on an older Mac. So the only way to run it these days would be on an old Mac or some kind of Mac emulator with all the right settings. But just very cool stuff. And you know, they had these different exhibits, and another hall just down the way was called Wild California. And again, giant dioramas. These are behind glass because they had suspended stuff. So you're supposed to look like things are floating or you're swimming through stuff. And they did have a blow up of the beach rack. And something like 500 times, or no, it wasn't that much. It was maybe 100 times, between 50 and 100 times. So you're seeing giant pieces of kelp that look like these huge structures, but they're really just very small in real life. And then all the isopods and amphipods and little shells that are found in there, and all kinds of other stuff. And blown up versions of the sand that you just saw in this that I showed you a little bit ago, in a massive diorama. It's just beautiful artistry. It looked very realistic, and I used to just sit there and just marvel at this frozen snapshot of time of these critters in the beach rack. And it's really hard to simulate that with this equipment, because you have focal planes and you have light issues and opacities and stuff. So it was really fun to see those exhibits. I miss stuff like that. I know they have to progress and make new exhibits, and again, this was like 25, 30 years ago, so of course it's going to change. But man, I missed some of those old exhibits. I wish I could revisit them, because they're such good quality, artistically built, and sound design in the exhibits with like insect sounds of the era simulated, because nobody really knows what they sounded like back then. But you can, based on what fossils you find and stuff, you can make extrapolations on the animals as they are now versus the ones back then, maybe scaled up or scaled down for the size and the changes in the bodies and stuff like that. But it's just, this is cool. You can just sit there if it's a quiet day, hardly anyone's there, and you just chill out in the exhibit, listen to the sounds, look at the scenery, and you really get transported to those places. It's really a special thing. So it's almost like armchair travel. You know, you don't really go there, but you feel like you're going there with the right setup, right information. I'm not seeing this guy anywhere, so he's either really hiding in the shady part of this under some junk, or he couldn't have expired that quickly. Let's just probe around a little bit and see if I can roust him. Come on, buddy. Just want to look at you real quick. I'll put you back in the bag. But it's significant magnification. You can see the forceps look pretty huge right now. Yeah, not seeing that guy anywhere. Yeah, AI could help. The problem with AI though is it's based on all the stuff I've seen and heard so far. Um, it seems to have some license of its own, so nothing really looks very realistic. Although it's hyper real, fantastic kind of stuff. It's gonna be a while, I think, before it gets to be not just fooling people with, Obama breakdancing and stuff like that, but to do this kind of stuff realistically, where there's a certain tolerance you need to have to make it look real, with the transparencies, the different lights. Yeah, I've done some, even the Mid-Journey and Dolly and all that kind of stuff. You can do pseudo 3D graphics on the fly with the prompts, and it's just basically faking the 3D. It's not really going through doing render passes and ray tracing and stuff like that. It's giving you the impression of a 3D image without really doing all that extra math. And the stuff's pretty cool. I wouldn't say it's as interesting to me anyway as a properly rendered image if you're trying to do 3D stuff. But it's definitely getting there. But this kind of stuff, it's so much detail and so many different textures and transparencies and color variants, and yeah, it's a much more complicated thing to do, which is why you don't see a lot of like Avatar 2, The Way of Water, has some stuff in it that's pretty amazing. Now that wasn't AI, that was just hard work of artists making stuff in 3D. There's some hybrid 2D, 3D in there for sure, but and obviously the actors are real. The human actors are real, just interacting with the replaced Avatar, Navi, whatever characters. But they even got the water simulations really well, and there's some scenes there that will blow you away where you see they have the fabric, like the woven ties on the 3D character's hand as they're riding those sea creatures, and the water washes up, and the way it wets the material and dissipates. The guys, there was a guy on one show talking about the effects, and he said he's claiming that was actually, that segment was all 3D, just a close up of the hand and the wetting of the material and stuff. And to have things wick like that in 3D, if you ever studied particle animations and fluid simulations, it's easy to do big waves and clouds and storms and stuff now, but little subtle details like wetting of a wicked object like rope and yarn and stuff, and just to have it look realistic, because we all know it looks like, you see it all the time. That's a hard thing to do, and they really pulled it off in Avatar 2. It's pretty much a crap movie, as you would expect. But, I mean, it's fun, it's adventure and stuff, but those little details are just, holy shit. Unbelievable. And I've been doing 3D for a long time, mostly just tinkering. I'm not that great, but I can at least appreciate it. It's just like the stuff, guys like Tom Savini and Stan Winston and Dick Smith doing amazing stuff with makeup way before CGI and stuff. And I can appreciate all that because I studied all their books and documentaries and behind the scenes stuff. And I've done a fair amount of makeup stuff myself. Prosthetic bits, not foam latex, that's too much work and too nasty, it smells. But with lots of other stuff, mortician's wax and liquid latex and flexible rigid collodions and all that shit. Most people don't know what the hell it is. And by just dabbling with that, you can really appreciate how much work it is before you even, put a lot of time into it. Because you get that from the book, you get impressed, and you get that from watching the movies. But then you really start playing around with it and realize how tough it is to get it to work out right, and getting materials to do what you want them to do in the real world because of temperature and other other kinds of conditions you're working in. So it's pretty wild. So when you see that stuff done that well, using the exact same materials you have right in front of you, they've not just mastered the art of the sculpting and proportions and things like that, but they understand the physical properties of all the compounds they're working with to make them behave and do what they want them to do. And that comes from a lot of reading, a lot of experience, and taking risks and stuff. So, all right, I give up on the little bug. I don't know where it went. What else we got in this bag? Not much, mostly some wet sand and a few other pieces of junk. So I'm going to give up on that. Let me take a few more little squirts out of it, make a couple more slides, and just see what we can see. I also was going to quickly look at some water I collected down in LA at Catalina Island. And it's been in the fridge this whole time, so there's a good chance some of the critters are still alive because they're pretty durable, and there's trapped air in there that probably used up by now, but I keep forgetting to use it. So I will try that real quick and see if anything can be found there. It was an ocean water sample and a rehydrated section of weeds and plants on a sidewalk. It was so dry in the island, other than the ocean, I couldn't find any puddles anywhere. So I had to make my own little sample with rehydrated water, rehydrated plants and dirt to see if it could either bring some dormant stuff to life or I don't know what I would find. We'll see. So there's one more. Let's just do one more sample from the water of the bag here. Let me switch the cameras again. And get this dialed in here. Let's see what we got here. It's very, very bright. Kill that brightness there. Way too bright. Sulfur's too bright. That's what it is. Okay, let's go. Again, particles, 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 but at this level I should be able to do dark field. Interesting. It's like a little cluster of red algae cells, maybe. Let's do a quick digital zoom to be lazy. Alright, what do we got here? So that when it's red algae, you probably noticed this before in other streams I've done where something looks consistently red or blue, but when you zoom in on it, it's really mostly white or something else, and just little elements of pink or red or whatever. Like blue jeans are mostly white fabric with some blue fibers going through it, and your eye fills in the details and makes them basically look blue with some white. Same with a lot of red stuff. It can start out pink. Like your blood, when you put red blood cells under the microscope, they're not bright red like it is when you look at it coming out of your finger. They're like these kind of reddish, pinkish color, faded, much, much more faded than the blood flowing out of your finger. but collectively, to your eye, it looks very, very red, even though it's more pink on the individual level. Let's take this a little closer so we can see. So yeah, I'm not sure what it's from. It could be from any of the red algae or something totally different, but it has the right color to be red algae. Cool looking. Again, no idea what it is exactly. Just a mass of cells. And there it is to scale. It's a little small bit. Looks like this cover slip wasn't cleaned very well by me. There's a bunch of extra debris on it. I did kind of a rush job to repurpose some of my cells. Ooh, is that a Nauplius larva? It is. Yeah, this is a copepod larva called a Nauplius. You already see the red eye spot. And crabs also can have a similar larval form. They're all related. And so you get these like little balls with bristly legs. And then usually this red eye spot in the middle. Red eye spot is right up here. A little dot right there. And these little legs coming out with little bristles all over them. So they have like sensation. It's like little hairs. And there's some stuff moving around it. It's not jerking or pulsing, or the inside's not really gurgling or anything, so it's probably dead. Or recently dead, so it hasn't started to break down yet and lice and spill its contents everywhere. Let's try getting a little bit closer without oil. This is the inspection music. It's very pensive and slow. Like, what is going on here with this Nauplius larva? There's the red eye spot. Here it looks a little more, it's more like it's carmine instead of like a cardinal red. It's more like a carmine. It's kind of this almost ruby color. It's more on the pink side than red. If I can get this balance a little bit better here with the light. Make it too bright. There's some of the contrast. So yeah, I can focus through the body. They're fairly thick. There's the bottom with the legs coming out. See all the little joints there. And as I focus through to the top, now you can see the top where the red eye spot is, near the front. But those little bristles there, you can see the bristles on the legs. They're not just little stumps, they're spiky. Little bristles on them. Really cool little guys. Very, very common in the plankton. You know, when you have crabs and lobsters and other kinds of stuff, they have a version of this Nauplius larva. This is just a simple, very small one for a copepod. But yeah, it's interesting little critters. But there's no activity inside the body. I'm not seeing anything moving or gurgling or pulsing or anything like that. And even when they're at rest, when they're not jerking their limbs around, you know, trying to swim or just trying to, you know, shake things off or whatever they're doing, there's still some activity inside. Right? This one looks totally dormant. But occasionally they'll surprise you. You look at it and go, hey, it looks like it's dead. All of a sudden, it takes off, you know? Pretty wild. So yeah, I like the Nauplius larva. It's Nauplius, Nauplius. Very cool. I'm not going to go any closer because it's such a big critter that it's just going to be mostly a blur with a few clear spots. But I might be able to adjust, get a slightly different viewpoint here. Maybe I can't. I can go back to maybe one more level and still get dark field. Let's see. Oh, here we go. We're getting some oblique lighting here. There. A little more of a 3D pop there. That's pretty cool. Let's do a digital zoom there without ruining too much of the detail. Still, see those little white particles moving around. This is all kinds of still gradients shifting through the water. Looks like it's falling due to gravity, but this is flat, so it's just some kind of a gradient that the particles are following. They're not things swimming. Oblique lighting there, kind of a 3D pop. So it's got some edge lighting on it, some dark edges and some light edges. Really makes it pop. I keep forgetting to do this, but I didn't have a lot of time to do this tonight. At least so far I've actually done for a while, but I need to do a whole stream on my phase contrast optics, which I still haven't used after I bought the scope. There's a box sitting there and I've not used the phase contrast at all. Give a very different look of this guy. Mostly better for small things like bacteria, particles of stuff, very small cells, makes them pop. For bigger things, looks different, but not always so great. Really depends on the object. All right, let's go back to zooming out a bit here. Let's do a quick survey of the rest of the slides. See what the hell's going on there. Before it dries out, it's already starting to crystallize again. Salt crystals are starting to gather. Let's go back to, yeah, this is good. Do some dark field. There's some more particle movement there. Above that cluster, going down to the left. Little particles streaming diagonally. Various cells of some kind here. Not sure what they are, but they're not doing anything. They're just sitting there. There's a diatom there to the right. This little rod there. Some more clusters of cells. Very big diatom there. It's a nice one. More junk, bits of clusters of diatoms. Zoom in on that a little bit. That's kind of cool. There's two stacked together. They're not conjugating like Paramecia do, but I'm not sure why they're doing that. Some of these get a little bit colonial at times. Get a few of them stuck together. I don't think they conjugate like this. I definitely have video, if you look through my, not the live videos, but the uploaded video section called videos. I have some footage of conjugating Paramecia. They're actually mating while they're swimming around. It's pretty cool. Don't see that very often. At least not unless you raise them, so you have them breeding all the time and stuff. Some more guys clustering together. These look like other diatoms here. These little round ovoid diatoms. That doesn't look like a diatom. Not sure what that is. Top. There's a really nice diatom there. It's almost, I don't think it's a Navicula, but it's beautiful. Look at that structure. It's got a lot of detail. Let me see if I can blow this up a bit digitally. Try and improve that image. And the eyepiece, it's beautiful, but a little trickier on the camera, especially with some digital zoom here. Let's try and clean that up a tad. It's got all kinds of structure inside of it. Really nice. Got some nice colors, some nice structure. There you go, some nice compartments there. Really cool. Nice. One of the nicest ones I've seen in a while. It's got that cigar shape. And they're thick enough that you can focus through them and see layers and details change. Basically little tubes, tapered tubes. And I believe those two blobby things opposite each other are, no, the oil drops are usually round. I don't think those are the oil drops. Not sure what that's just part of the photosynthetic structure there. Really nice though. Looking at your, sorry, I'm behind the messages here. They have just one eye. In the Nauplius larva, it's a compound eye. So it is one eye spot, but it's made of various cells. So something like a spider, they might have these little things called ocelli, these really simple eyes, just a little dot. It's essentially just a equivalent to like a photocell, just a single point, big or small, depending on the organism. But just gives you an image of the area in like brightness values, no sharpness, just overall, like they see if it's getting dark, something's probably coming at them fast, right? If it's too bright, oh, I gotta stay out because I'm visible, right? I gotta hide in the shadows to be safe, or not to dry up or burn up or whatever. So that's the simple eyes, and then you get something like a dragonfly, big old compound eyes, and multiple cells. And they don't just have compound eyes with the multiple cells, but depending on, you get this with praying mantises a lot too. The eye can actually internally change the way the light is funneled through the eye. So when it's dark, it can actually collectively use the cells more like a bunch of simple eye spots, and when it needs to be active and light and it's hunting and needs to focus really well to attack the prey, the eye can actually change, so it's now acting more closer to how our eyes work without a lens and without all that complex, internal structure. But you can change. There's a really good article in Scientific American I saw like 25 years ago that shows how the eye can actually change based on light levels and behavior of the animal at the time, hunting mode versus like gather as much light as possible in its dim area for security, for saving your butt. And really fascinating they can do that with something that doesn't look as complicated as a mammalian eye or a bird eye or something like that. So in the case of the Nauplius, it's just a single eye spot made of a few sensors clustered together, like primitive sensors. But eyes are really fascinating. I mean, I not just that our Scientific American article, but the textbook I was using at the time, and these eyes of origin of eyes in embryonic development in different animals. It's fascinating. So our eyes grow out of the neural tube tissue as we differentiate. So you have the blastula, which is the ball of cells, and then it hollows out and makes a gastrula, and then it starts to redefine itself more and get compartmentalized and get your symmetry. You get bilateral symmetry, radial symmetry, whatever your body's going to be. In our case, it's bilateral symmetry. And as you elongate and form different cavities and structures, you start forming this notochord and a neural tube. Notochord becomes your spinal column eventually, and the neural tube is exactly what it sounds like. It becomes your spinal cord. Starts as a primitive neural tube, but something like a tunicate is actually a relative of ours because it's considered a chordate. It actually has a notochord. It has a very primitive, at least a notochord. I think it has a very primitive neural tube as well. I kind of forgot some of those details, but it's in chordata, so it's distant relative of ours. It's the tunicate. It's little called sea squirts. These little gelatinous things in the ocean that just siphon water in and out through these different siphons, and looks nothing like anything that would be related to us, but it's at least a chordate. It's a phylum called Chordata. So they've redone some of the taxonomy since I studied it heavily, but now we have domains at the very top. So this domain is I looked this up recently because it's some of it's a little bit new to me, just because I have it hammered in my head. By kingdom, right? We had kingdoms. So kingdoms was Animalia, Plantae, fungi, one called Monera, which covers all the bacteria, and blue-green algae, and all the basically prokaryotes. They don't have a true nucleus for the genetic material. And then you had this one called Protista or Protoctista, which is all the catch-all for everything else, including the algae, because they start as a single-celled form for the most part, and then differentiate into all these structures, depending on the type of algae you're looking at. So it was called a five-kingdom system, and over time they started splitting and changing this and that, and hemming and hawing and stuff, and then they decided to make it more, go even upper level from that, because you want to now classify back viruses as a life form, even though they're purely parasitic. They cannot live on their own as a life form. They're just genetic material without a host. So now they have their own domain above kingdom, and I forgot what it is. I have to look it up. But and it's late. I'm getting tired. But basically, domain is the new top level. So it's not just the kingdom down, it's domain and then kingdom. But for example, humans are, so we're kingdom Animalia, phylum Chordata, subphylum Vertebrata, order primates or primates, if you did the true Latinized speech. And then after that, it's we're in the class Mammalia, order Primates. I got it wrong, sorry. It's been a long time. So basically, kingdom, phylum, class, order, family, genus, species is the order going down. And of course, domain on top of kingdom now, but I forgot to call the domain. So, kingdom, Animalia, phylum, Chordata, subphylum, Vertebrata, class, Mammalia, order, Primates. I think it's there's a superorder in there, I think, somewhere, or no, a suborder, whatever, for some more primate stuff. Because people say, oh, we're not primates, we're humans. It's like, no, we share DNA incredibly close to the chimpanzee. We're fucking primates. And then class, order Primates, family hominids, right? And then, genus Homo, and species Sapiens. And if you want to get really technical, they often throw a subspecies there, Homo sapiens sapiens, because I think they found another subspecies of Homo sapiens through some research a couple decades ago, but I haven't kept up with that. In general, it's just genus species. So like, the wolf is Canis lupus, genus species. The chimpanzee, Pan troglodytes, right? So it's just a bunch of different genus and species out there. So nice thing about the taxonomy and using Latinized stuff and the whole world agreeing to it, the scientific world, is I can go to a guy in Japan and say, oh, Canis lupus. He doesn't speak a word of English, but he knows what Canis lupus is because he learned the taxonomy the official way that the world has agreed to. So it makes this kind of universal language where you can read a paper and then it's going to have all this kanji in it potentially, and then you see in parentheses with italics Canis lupus if they're talking about wolves. So that's very cool, and that's actually been able to use even Japanese sites on microbiology to help me find some critters sometimes I couldn't identify. Some really good websites out there, because very few books on this stuff that are very useful that aren't really expensive. So they're great websites all over the world with this imagery and with the names and stuff. So if you just have time to quickly do a visual search and not go through some kind of huge dichotomous key to key out something with all these characteristics, they're sometimes hard to find. You can use a type of image search and getting ballpark at least. You can at least maybe figure out the family of the order of the certain type of thing. Everyone knows, Paramecium, caudata. It's very, very common. There are some other ones out there, but that's the most common throughout the world. So most likely you're going to find one you can tell very specifically by the size, the shape, and certain other attributes. It's not just Paramecium, it's Paramecium caudata most likely. But at least you got close to the genus, you got Paramecium. But a lot of other things you got to go by, oh, it's probably this family, like Amoeba proteus is another famous one. There are lots of amoeboid things inside of shells. Like I have a heliozoa thing I did several months ago, where it's a heliozoa being kind of bombarded just in passing by a bunch of gastrotrix. And the heliozoan is a single-celled amoeboid inside of a glass shell that looks like a sunburst. And here are these gastrotrix swimming by, and they're multicellular, but they're almost the same size. And they're 1,000 cells versus one giant cell. So this stuff's all crazy. It's all kinds of stuff. But the taxonomy is fun though, because again, it helps you keep things compartmentalized, figure out relationships. The taxonomy changes as they find new genetic differences and similarities. And again, it's a universal language. I can talk to somebody in Germany, somebody in Japan, somebody in Iceland, and use the scientific name and they will know what the hell I'm talking about. Common names are a mess because they're regional, they're based on the local language. English common names for some of the plants and animals are not only ridiculous, but they're regional. So you might have like 10 or 20 of them for the same plant in the US because it's regional names. Also, some of them based on the old days, like the whole "Let's make America great again" when things were even more racist. And there's some horribly racist names for plants that I won't repeat on the stream. But if you look at an old flora book of California and other places in the US, they had common names for most of the plants for people who are just learning. They don't want to learn the scientific names. But they have these regional common names. And they're very, very unpleasant because they're racial and racist. And yeah, so that's enough said about that. I'm not going to go to the names. But it's pretty awful stuff, you know. Yeah, it's easy to forget all this stuff, Matt. I mean, I've been doing this shit long before college, and you know, got my degree in this stuff. Not microbiology, but you know, molecular biology and zoology and botany. But mostly on molecular biology because that was what the degree was. But all my self-study and all my hardcore interests outside of class were ecology, plant-animal relationships, plants and animal taxonomy, identification, all kinds of stuff, and you know, habits, natural history, all that stuff. But the amount of stuff I've forgotten, and it's not a brag, it's kind of sad because when you're younger, you're just a huge sponge. And for a long time, I was a walking encyclopedia. I could do tours of the botanical garden I worked at, or the zoo I used to work at, and just spout off endless stuff. That wasn't just something somebody told me. I synthesized a bunch of stuff from books and documentaries and classes I took, and some lectures, and friends who are well learned about stuff, and synthesized my own kind of spiel about stuff. And I could just go around and just talk for hours about this stuff, and I loved it, and I still love it. But most of that stuff's gone, man. You get older, you learn new stuff leaves as you don't use it, or you just got to make room for other stuff, and it's frustrating, you know, but it's life, and it's part of getting old. And so I'm glad I can remember what I can, but when I think back to what I forgot, it's really depressing. So I just keep plugging along, and every once in a while you get a little, ooh, I forgot about that. That's right. Oh, and something will trigger it, and then all of a sudden I remember all this stuff about something. But then it could be somewhat fragmented because I'm remembering chunks of it. And so it's always a bit frustrating, but you know, when you get in the groove and it's a good, you got enough sleep and you got enough energy and you're not taxing yourself too much, you can get in the groove and just remember all kinds of stuff. And you know, people who are into that stuff appreciate it. You know, they're not like people who just want to remain ignorant or say, "Oh, that's not true. I read a thing, some conspiracy theory about this." I'm like, "Oh, all right, yeah." I have no time for any of that nonsense. I have enough trouble having time for the real stuff that actually matters. But you know, like you're saying, art and artists and stuff. I got into art a lot in college and before college, and after I got more into history than I was in high school. I got more into you know, geography, and it's just your head can easily be just full of stuff, and you're like, how am I going to fit more stuff in there? And then eventually you forget some of it and go, oh, suddenly it's easier to learn some new stuff because I've already forgotten some stuff. Although that's not really how it works. Feels like how it works sometimes. But yeah, it's pretty wild. So, and this is another reason why, whatever you have, hobbies or interests or keeping active and diversity and learning new things as much as possible to keep your brain plastic, so you can keep forming new memories and holding on to memories and stuff. My grandparents, especially my paternal grandparents, as they got older, just sat around watching TV, soap operas, game shows, and it's just, the brain just rots, right? My grandmother on my dad's I was a teacher. And she used to do crossword puzzles. She was damn good at them. So that's one thing she did up to a certain age where she was keeping her brain going at least, right? But even that, she stopped doing that, or probably had trouble doing it at some point and just vegged out in the TV. And so bad, I go over there sometimes and my grandparents are talking about characters in the show like they're real people. Can you believe what Bill did with Sally? And it's like, what the fuck are you guys talking about? These aren't even real people. Because they don't get out as much as they used to. They just, kind of veg out, and stop reading books, and stop, this is before the internet mostly, so they wouldn't know how to use a computer if you put it in front of them anyway. And they just don't have a zest to learn anymore. Just want to relax and kind of slowly fade away. And it's, man, I don't ever want that to happen. Obviously, if I have a traumatic brain injury or something, I have no choice. But as long as I can, think and synthesize ideas, and I'm gonna keep plugging along because it's just what I like to do. It's my whole life. But also, it's very important to keep doing that, whether you like it or not, to keep your brain going. But yeah, I mean, yes, synthesis is, master FM synthesis. You're keeping your brain going. Or any kind of synthesis, but especially weird stuff like FM synthesis and additive synthesis. You ever try to use a K5000? Holy shit. Anyway, enjoying this little diatom for a long time, but it's a beauty. It's like a little yellow gem. A little yellow-green gem. There's a little guy next to it with all kinds of little bacteria here. I don't think that's a diatom, but it actually might be a diatom. The color and the structure might be one of these other weird diatoms I haven't seen before. Because this is from the ocean. And most of the stuff I collect tends to be from the salt marshes, which is like estuary. You get a mixture of fresh and salt, or freshwater. Helps to defocus sometimes to get some details to pop and colors to change. Ooh, that's a nice diatom there too. And all kinds of little bacteria floating around it. Little black dots. Yeah, so there you can see the oil drops. See those little spheres in there? Nice little perfect spheres. Well, they look like circles, but they're actually spheres. Those are oil droplets. And it's been a while since I read about those, but if I remember correctly, they're partly food storage and helps them stay at a certain point, and it helps with their buoyancy in the water, so they stay at a certain level and don't have to fight. But again, it's the cobwebs are coming out of my brain right now. This is a cool one below it. It's more like a cone shape. Look at that one below it. So sometimes you get a nice tapered cigar, sometimes you get a cone shape, you get a sphere, or a pinwheel. I have a really cool one I shot. Oh, sorry, I'm off camera here because I zoomed in. This is the cone shape one, and it's swimming. A little hard to see the boundaries on this one. It's not very high contrast. But if I focus, you can see the boundaries. But this one's stunning. It's another nice one here. I love these guys with the kind of cigar shape. But yeah, you can see those little oil drops, those little spheres. There's a couple right here. Boom, boom. And there's a couple here. Boom, boom, boom. Those little spheres right there. Those are oil droplets. And I need to read up on those some more. I forgot some of their significance, but some are really prevalent, and other ones you almost never see for whatever reason. I don't know. So in this case, at least from this side, or this point of view, because it is transparent, I'm seeing 4, 5, 6, 7, maybe 8 oil droplets. A couple different size ones. 1, 2, 3, 4. Looks like 5, 6, 7, 8. At least. And it's funny because even when we studied these in lab, because so many things to study, you kind of gloss over a bunch of stuff. So we didn't go into the specific details in the diatoms of their type of chlorophyll or the differences in their photosynthetic structures compared to some other single cell plants. And maybe oil drop was mentioned, but no one ever really talked about it that much. So it's one of those things. Need a lot more self-study. What else we got here? Another one of these guys. Again, might be another weird diatom. Not sure. Could be just another single-celled algae. But that looks like a very clear shell, like a diatom, instead of just a cell membrane, cell wall kind of structure. More clusters of junk here. From something, looks like something fragmented. There's that pink thing again. Alright, I'm going to zoom through the rest. It's starting to dry out, I think. See what else we can see. What else can we see? I can identify at least. So there's a big old diatom there, one of the boxy ones. Little oil drops all through there. Oil droplets all over the place. See if we can get a different viewpoint here. Yeah, I don't have the right patched up in there, but lots of oil drops there. It's a little rounded diatom there. More of these guys again up here. Well, that, at a glance, that could be an amoeba, but it's not doing anything, so I'm not sure what it is. But it looks like it has a nucleus. Not seeing any pseudopods or anything. Ooh, there's all those little tapered diatoms. So little skinny little, way out of defocus to see them, they're so skinny. Long tapered ends. There's a piece of something here, look at that. It's a little hooked bit and some little spikes on it. Not sure what that was from exactly, but it's definitely a piece of something bigger. It's almost like a giant, like a much larger Nauplius larva. One of the legs or something. More of these little guys. That's definitely got diatom structure to it there. Don't know what that is at all. Looks very diatomy. else we got? More junk, more junk, more junk. More diatoms. There's got a shitload of diatoms in this sample. A lot of these little... Oh, here's a piece of filamentous algae. Like, could be even a blue-green algae. Like Anabina. Oh, that could be something significant, but I have no idea. Definitely well contained, whatever it is. Reddish color. Diatom, diatom, diatom. Ooh, is that what I think it is there? I think we've got some spirilla. Yeah, see that little, to the left, it's kind of transparent. It looks bent. I believe that's a spirilla, one of the spiral bacteria. And when it gets going, it has flagella at different angles, and it can spin. Like a corkscrew, it spins. And it goes forward and backward in a line when it feels like it. And with that spinning behavior, it can easily tunnel through like masses of material and gelatinous stuff. Instead of being a blunt thing, just trying to butt its head on something. That spiral motion they figured out through evolution so they actually have proper spirochetes, which have multiple turns, like fusilli pasta. But then they also have sometimes only one turn, like this one, or 1.5 turns. And it looks like almost like a peanut, but bent. And when they start spinning, it's really cool. Right now it's just kind of bumping around, it's not spinning. But I see them a lot. Very characteristic bent shape. And not super long, they're kind of short. So this isn't even the highest power, this is 400 power, and you can still see bacteria quite well. You don't have to go to 1,000 power to see them. There's a better shot of them kind of wiggling around. Some of the kind of bent stubby ones, those are like spirilla. I've never seen the long spirochetes, the really long fusilli pasta ones. I'm not sure how rare they are, but I've never seen them. You basically have rods, spheres, and spirals. Those are the three main types of bacteria. And they can be colonial beyond that to make bigger structures. So like E. coli is a rod, it's called a bacillus. And it's just like a cold capsule you would take compared to like an aspirin. It's instead of a round flat tablet, it's an elongated tube with round ends. So that's a bacillus or rod. And then a cocci is the plural for coccus, is a sphere. So like someone called coccidium something or other. It's often used as a naturopath remedy, whatever. It's supposed to help you with some shit. A bunch of things that have cocci in the name, C-O-C-C-I, and that's spherical. Like Staphylococcus is like Staph aureus, the famous infection you get. It also can be flesh-eating bacteria. It's pretty nasty. Or MRSA is another version of that stuff. Something resistant Staph aureus. I forgot the M. And it's a really nasty. You get flesh-eating bacteria start dissolving your arm, and you basically often lose it if they can't stop it. So Staphylococcus is the spheres, and then spirilla is an example of a spiral structure. So, cool stuff. Just seen a lot of the same stuff, nothing new. Some more little diatoms. I may put this water in the fridge and do something else later. I'm getting tired, but whoa, look at that. That's a big structure. I gotta zoom out for that. This digital zoom is too big. Look at that. It's almost like a primitive hand made out of cells, but little tendrils. But look how it branches out. Very interesting. Get that stupid hand out of there. Really cool. Really, really cool. So it's big. It's actually going off screen here for me, and even in the eyepiece. And there's a nice little diatom next to it. A little oval thing with a little. That I'll zoom in for again because that's pretty cool. Oops. This guy right here. Oops. Wrong control. See, as I focus through, you get the little compartments. That looks like the rafe, the little slots you get in the diatom shell. Lots of weird tissues and pieces of things floating around. When you consider this is the beach rack put in watery sand in a bag, a bunch of stuff shook loose. So you're seeing stuff that's, oh, beautiful. Look at that one. Nice diatom. Again, slight variations in the shape. Pointy, blunt, tapered, not tapered. Some are rounded, boxy. Lots of bacteria in this, around this guy too. You see him dancing below it. I'll defocus a bit so you can see him dancing. Really cool. Structure there. A lot of tissue-like things here. Things are breaking off of things. Hard to imagine what they go with. There's another little cigar diatom. Right there. Ooh, there's a nice boxy diatom. Boxy but rounded. Light to that. Zoom in a tad. See a little bit better. Getting some water shifting. Looks like the slide is starting to dry out. That's when you get those little pulses of water. So lots of little oil droplets around the outside. If I defocus them, you can see little spheres there. Very cool. Pretty cool. Oh, those little conical diatoms. And the long pointy one. Bilaterally symmetrical. Right there in the middle. Nice diatom, very nice. It's like a kayak or something. So yeah, nothing crazy so far, just some interesting, typical stuff. Going off. Slide there. There's a nice conical, looks like a dunce cap. All full of goodies. Full of goodies. Plenty of oil droplets in that one. Alright, pretty cool. Let's find one more little diatom. There you go. Ooh, this one's got a little color in there. Look at that. If I didn't know better, I'd say it's an eye spot, but they don't have eye spots. So it's either something right on the outside of it, or it's actually inside. Yeah, it looks like it's inside the shell. This color, there's a little, look at that little bent. Oh, actually not bent, there are a couple of those bacterial rods there bouncing around. Just to the side there, going down to the bottom tip. Bouncy, bouncy, bouncy. So in phase contrast, they would just pop, you wouldn't miss them. Here I have to defocus a lot to show them because there's really just little outlines with a white background. They're very hard to spot unless you know what you're looking for. But you cannot miss that diatom. It's a beauty. Just funny with that red spot there. Anyway, I would stay longer, but I have to get some sleep. Another crazy day with wedding party part 2. If you got married today and now there's receptions tomorrow, it's like, can't you have them on the same day? But hey, it is what it is. I'm just along for the ride, so. Won't be by the ocean this time, don't think. So won't have any new samples, so that's why I want to get it done today. You know what? I'm already set up. Give me a second. Let me go grab the tubes from Catalina. Maybe there's something in that water. I'll do maybe 2 more slides. And oh, thanks, Matt. Yeah, hang on a second if you're still around. I'm going to do Catalina real quick just to see. I'll keep the dirt slide in the fridge because the dirt will be interesting for a long period of time, I think. But the water will probably expire because it's if it has protozoans and stuff in it. Hold on a second. Okay, let's see what this does. Catalina Island samples. They're still viable. So this rehydrated plant soil sample I did is very colorful, so that should be interesting. This is very plain looking. It's just ocean water taken as I ran up to the wave and ran away before my shoes got soaked. Let me quickly rinse out this pipette so I don't contaminate the sample. Yeah, it'd be nice to go back to Catalina or places like that when there's some rain going on, so you get actual puddles and be all kinds of cool local critters growing in the puddles, fresh water. I drove around the whole island in these little golf carts because the island is basically tiny roads with very few permits for locals to have any real cars. So it's little tiny Scions, little tiny smart cars, little Mitsubishi Fuso minivans from Japan. You see all over Japan if you go there. But it's funny to see a whole bunch of them in an American island because they have perfect size for the island. Just quickly rinsing out my sample. Quickest way is actually use some isopropanol. Basically denatures everything inside eyedropper. Without having me go to the sink and flush everything out. And I just flush it with a little bit of drinking water here. So I don't kill whatever I take into it, because alcohol denature whatever goes into the pipette. Alright, that should be pretty good. Okay, I think that's pretty good. Should kill whatever's there. Just put a little bit into the cap. Sample that. Give it a nice little shake so I get some distribution. Put a little bit on the slide and see if there's any magic. So this is ocean water right off the beach at Catalina Island. I better start with some lower power first, of course. Let's do a little dark field to start. Makes it a little easier to find stuff sometimes. Open that up a little bit. Okay. Too much zoom. Not enough light. Oh. Let's see what we can see. This edge of the cover slip. It's the top of the cover slip. I want the stuff underneath. Yeah, I should clean these cover slips better. They're dusty. Little fibers. It could be from any old thing. Special report, some little pieces of junk. nothing worth zooming in on. Let's try one more. Same slide, same sample. Cover slips. Put on my cover slips. Got a couple more. Goody goody. Alright, so it's on the side of the attempt number 2. Wow, cover slips. Smudgy. But I don't really see anything there. Now, of course, these samples are now like a week old almost. So, even though they've been in the fridge, life hasn't stopped, so that things could consume most of the resources and just die. I've seen a lot of little bits of stuff. Could have been anything. Alright, well. That was a wash. But that's what I get for waiting too long on that sample. Let's try the rehydrated sample from... What the hell? I dropped my... Oh man, I dropped my cover slip on the ground. Shit. I think I saved it. Alright, let's put this cover back on. I don't want to mix the marine and fresh. Let me flush this one again real quick. I haven't done this for a while and I misplaced my little bag of pipettes, so I have to keep using the same damn one the whole show, which is not ideal. So you want to compartmentalize your tools so you don't cross-contaminate. That's flushed enough, I think. Let's try another slide. Okay. Let's invert this so I get a little mix of whatever. Nice suspension in the liquid. Yeah, it's anyone's guess what I'm going to find. It's kind of silly actually, because it was so hot and dry there. Again, except for the ocean there. I got the sample just before I left to catch the boat back to the mainland. I just poured a bunch of my bottled water into a nice little sidewalk crack that had some dried up plants and dirt. I poured the water and let it sit for a bit, and then I used a pipette to transfer a bunch into a thing, and then dumped a bunch more water into it to dilute a little bit because it was could get so opaque you couldn't see anything with it. Plus, it had to rehydrate a bunch of stuff, so much of the water was going to disappear anyway into the tissue. So let's see what I got. See if it worked. Had a nice color to the water. Could just be the dirt particles. Could be some good stuff. Some tissue, plant tissue. It's kind of odd, but different. Let's go back to lower power with some dark field and see what pops. As I suspected, little patches of plant cells and some root bits and Alright, let's do back to low power. So we've got pieces of tissue, various kinds. Some green, some brown, some whatever. Oh, I gotta fix this. My condenser is not. There we go. Okay. Some little reflections of my that's better, I think. Go. Okay. Tissue, tissue, tissue. It's very colorful. That red is really hard to capture. I gotta blast it with light. So there won't be any diatoms here because, well, there shouldn't be because it wasn't a place that was supposed to have a lot of water. Lots of colorful tissue bits. It's like a seed cover, like a seed, either a small, like could be a grass flower, bract or something like that. Hard to say. Lots of root particles and such. squiggly bits. Yeah, that's all there is to it. Something different to look at. It was a last minute decision to try and find out some kind of terrestrial sample, and it just didn't work out. So, it just goes to show you never know. So, and I just ran out of cover slips anyway, so I'm going to call it there. Thanks for hanging out, Matt. Anyone else who comes along, watches the after stream, make comments, whatever you need to do, be appreciated. Always good to hear some feedback from folks. And more goodies next time. Try not to make the break so long next time. I literally haven't done one of these micro streams for, God, it seems like a few months, at least. So, and again, I got to finish with the JX3D music as usual. So, go check out his stuff. He's probably gonna have another senior stream coming up next month. Be a big surprise what it is. I never know until last minute. So, that's about it. Thanks everybody, especially Matt, because I think you're the only buddy here. And always appreciate to have some live viewers. I know I do this last minute at a weird time, but it is what it is. So, gotta go sleep. Thanks again, everybody, especially Matt. See you later.